Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled AAV Capsid Analysis and Quantification with Chromatography and Ultracentrifugation-Based Techniques. I'm Charlotte Barker, an editor at BioInsights, and joining me today is Andrea Livk and Sebastian Pellan, who will cover multimodal detection techniques and improved anion exchange chromatography for precise empty-to-full AAV capsid ratio determination. After the presentation, we'll have a live Q&A session, so feel free to pose your questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to those as many as we can during the session. If you can, let me know who your question is directed to. If it is to one of our presenters in particular, that's very helpful for me. I'd also like to draw your attention to the resources on the right-hand side of the screen, where you can find out more information on the topics covered today. So now I'll just briefly introduce our presenters. Andrea is the Head of Process Analytics Development at Sartorius BIA Separations, specializing in innovative chromatographic methods for analyzing biological molecules and with expertise in protein chemistry and biopharmaceutical characterization. Sebastian is the Head of Basic Research at Sartorius BIA Separations, leading the development of monolith-based applications and post-DGUC profiling for biomolecule characterization, with expertise in chromatographic purification and in-process analysis. So without further ado, I'm going to hand over to Andrea to kick off today's presentation. Andrea, over to you.
Hello, everyone, and a really warm welcome from both of us to all who are joining today's webinar. So our talk is divided into three parts. First, I will present how organic modifiers improve AAV capsid separation. In part two, Sebastian will discuss about the density gradient ultracentrifugation, its concept, and how this technique applies to quantifying empty and full capsid. In part three, Sebastian will also address spectroscopic properties of AAV capsids and response alignment between different detection modes. So AAV is the recognized leader in gene therapy due to the high potency, ability to target specific tissues to precise delivery, stability of capsid, which is easier to purify, and additionally, multiple approvals have been granted by FDA. During the AAV production, not only final product full capsids are generated, there are also empty, partially filled, overfilled aggregates and more, which are like protein-related impurities generated. So characterization specifically of these product-related impurities is still challenging. There are a broad range of different methods available for characterization, like analytical ultracentrifugation, mass photometry, all these 96-well plate assays, CDMS, and so on. However, generally, all these methods display low throughput, they are time-consuming, they require product concentration, high product concentration, and purity. They are less suitable for in-process or release testing in QC environment. So today we present two techniques. One of them is analytical ultracentrifugation. One of them is ion exchange chromatography, which is a very well established technique, specifically demonstrates scalability, modularity, and a high throughput analytics, and a density gradient ultracentrifugation. For both these techniques, we used multiple detectors, and we also, at the end, we aim to find out which of the detectors provide us the most reliable result. So improving for improving AAV capsid separation, that was really developed on strong anion exchange monolithic columns. So just a few words about the monolithic columns and why they are optimized for purification and for the analytics of large biomolecules. Monolithic columns, they are made as produced as a single piece of polymetacrylate monolith material. So they are uniform monoliths. They have channels, not pores, and all this interconnected network of the channels. There is really no depth, and ensures really enables low back pressure and high flow rates. Monoliths are structured in a way they offer convective mass transport, so which ensures low independent capacity and resolution. Functional groups which are sitting specifically on the surface of these channels are easily accessible to large biomolecules, which is resulted in high binding capacity. Monoliths, they are pore-free structure. They have low carryover, and they eliminate diffusion limitations. So we have different chemistry modalities of these columns, and they are ranging from small scale from 1 liter to 8 liter scales. So for these separation strategies, we use quaternary amine monolithic column. So we took the reference method where the empty and full separation was achieved in increased linear gradient. As you see, the poor resolution between empty and full separation is obtained. However, when we applied organic solvent, different organic salt and magnesium ion, much better resolution we obtained, specifically between empty and full capsids. But not only that, in the next few slides, I will show you that we started to see also other AAV subpopulations and species. For all these strategies which we used, I will now individually address in the next few slides and how they really impact on empty and full separation. So first, we performed various organic solvent screening. The concentration of the solvents was 2.5% in the loading buffer. And these various organic salts, they are differing in their physicochemical properties. Mostly, as we can see, the organic modifiers increase or improve the empty and full separation. This is the chromatogram in the middle where there is no organic solvent added into the buffers. And then the best performer what we obtained was acetonitrile. So acetonitrile was then selected for the further studies. We also selected acetonitrile, there were also another two reasons. Acetonitrile is miscible with water in all ratios, as well as enables decent solubility of various salts. So what we did next, so we did screening of different concentration of this acetonitrile, and so different concentration of acetonitrile. And what we observed with the chromatograms, the concentration of acetonitrile influence on the conductivity, elution time, and also the peak shape. At high concentration of acetonitrile, 10%, 20%, the peaks are eluting earlier, what we observed, and also we are obtaining sharper peaks. But not only that, if you look at the chromatogram with, more than 20% acetonitrile, what we already observed is also the appearance of this peak in high war salt. And this is some kind of quite possible that protein degeneration appears at that percentage of acetonitrile. The significant improvement between empty and full separation was achieved with the lower concentration of acetonitrile. Specifically, 2.5% acetonitrile was really the optimal concentration, which we then went ahead with further testing. So here we performed different salt screening. So we tested magnesium and calcium salts combined with soluble organic anions. So we know from the literature that magnesium chloride has positive effect on the specifically empty and full separation. But when we compare this magnesium chloride salt with the acetate, organic acetate anions, we see that much more better resolution was achieved. When we compared different organic anions, let me see, acetate versus formate anions, we obtained again with much better resolution was achieved with magnesium acetate ions. So we can conclude from that organic anions improve the solution, and the optimal anion was acetate anion. Also, then we tested different concentrations of the acetate buffer, magnesium acetate buffer, and what we see that at a higher concentration, peaks are eluting earlier, which appears really that they are less favorable binding conditions. So optimal concentration for that for the salt screening was less than 5 millimolar. So then at the end, when we as we obtained the buffers and the method, we applied this method also to other serotype, AAV9 serotype, AAV 2x8, and AAV 5 serotype. So on the top of chromatograms, I'm showing you a reference method. Here we monitored fluorescence as well as UV detector. And what we are seeing is that and on the bottom we have optimized method with these optimized buffers. And what we are seeing that similar elution profile we obtained for AAV8 and AAV 2x8. For AAV5 serotype, we see that peaks are eluting closer to the gradient, but still we managed to achieve the resolution. For AAV9 serotype, there was no resolution obtained with the reference method. These are all linear salt gradient. However, with the optimized method, we really nicely see the empty peaks as well as full peaks, which is really indication from UV to 60 to 80 detector. However, this method will slightly need to be optimized, but anyway, really the full capsids also there is not symmetrical sharp peak. We see that other population of AAV full capsids are present in the sample, and we know that also that AAV9 serotype really it has the most PTMs, which also probably reflects in this heterogeneous of full capsids. So I previously already mentioned, that fluorescence and UV detector, but really the method which we performed the screening, really we used triple detector, that is fluorescence detector, where we really monitor tryptophan intrinsic fluorescence of capsid proteins, and then the you will see a little bit later that percentage, the response on fluorescence detector correlates directly to the empty and full concentration. For light scattering detector, this detector really will provide us, is used for determination of capsid size, also mass, but for the, it's also very useful, this or almost like a mass to have detector for specifically when you're working with more like crude samples, not so much purified samples, where this detector will work as selective detector. And primarily we monitor UV absorbance detector at 260 to 80 nanometers, which is really estimation of empty and full capsid proportion in the given peaks. We also then performed the method validation. So we did a linearity, a limit of detection, and limit of quantification. So on the right-hand side, I have the calibration curve of for the full capsids, which is blue line, and for the empty capsids, which is red line. So we performed this parameters for fluorescence detector, for UV 260, UV 280 detector, and for the light scattering detector. So as we observe from these calibration curves, we see that they're differing in between the detectors, also between the full and empty capsids. That the biggest difference between these calibration curves or slopes of calibration curves is on the UV 260 to 80 detector, and also then to the light scattering detector. The slope for full and empty capsid is it's almost like the same, So, and as I mentioned before, really fluorescence detector correlates or response on fluorescence detector correlates directly to the actual ratio of empty and full capsids in the sample. For the UV 260 and 280 and for light scattering detector, correction factor are needed for the full capsids. The linearity we took we look at in the concentration interval 1 exponent 9 to 1 exponent 11 vector particles, and really primarily signal which we used for the quantification was fluorescence signal. So what we did also next, we performed also, we tested also robustness of this method. We tested it on seven different columns, as well as these columns. They're coming from they came from 4 different column batches. And as we nicely see also from the table, that all these relative standard deviation were below 5% for the conductivity elution, for the relative fluorescence area, and also resolution between empty and full capsids. So we also did the next experiment because we wanted to see how this method is scalable to larger preparative levels. And on the left side, I'm showing you chromatogram for the analytical method, which was performed on 0.1 milliliter volume of the column. And on the right-hand side is completely almost like identical elution profile of empty and full capsids on the 1 ml column. But this, monoliths really enable method scalability across various quaternary amine, high reproducibility column line. So this line was released last year, and really with this line we achieve high reproducibility, high resolution, scalability, and productivity of the column. So if I want to conclude at least the first part of my talk, the improved ion exchange method, which was based on and developed on the quaternary amine high reproducibility monolith, really enables baseline resolution between empty and full capsids. But not only that, we started to observe additional AAV subpopulations and species. The method was developed using organic modifiers, magnesium ions, and organic anions. So we applied simple linear gradient. The method is applicable for various AAV serotype. With method validation, we can conclude the method is robust, reproducible, and linear, with a low limit of detection and limit of quantification. The method is also scalable, which is really a key advantage specifically for AAV purification. And also the improved ion exchange method, some kind of established foundation really for future research and development between empty and full capsids. So now I will hand the floor to Sebastian, to give us more detail about the density gradient ultracentrifugation.
Okay, so thank you, Andrea. With the next few slides, we are stepping away from chromatography for a while, and we'll have a look after the new approach that we have developed in our laboratories. So the new approach bases on density gradient ultracentrifugation, which is a well-established purification method that enables separation of large set of biomolecules. So when we do ultracentrifugation, the high rotational speeds are implemented to achieve high gravitational force. And under the influence of this high gravitational force, the biomolecules sediment. Simultaneously, during the centrifugation, also the gradient forms, or sometimes the gradient is performed ahead. But the most important is that in this density gradient, the molecule sediment, either by differences in molecule density or by differences in sedimentation speed. And as a technique, the density gradient ultracentrifugation is orthogonal technique to, for example, chromatography. So here, as I mentioned, the molecules are separated according to according to density, whereas with chromatography, the molecules are separated according to size, charge, or, for example, hydrophobic nature of the molecule. Density gradient ultracentrifugation is, as a matter of fact, quite versatile technique. By implementing different density gradient media, density gradient types, and density profiles, it is possible to separate great selection of biomolecules. And before the advent of chromatography in 70s, the method was actually used for purification and separation of viruses, bacteriophages, EVs, DNA, and even proteins. So after the centrifugation, the material or the sample is retained in a closed centrifuge tube. So a dedicated protocol is required to extract the sample from the tube. The protocol is briefly described here and involves inserting the needle at the bottom of the tube for the drain. Then we insert another needle at the top for the venting. and when we insert the top needle, the liquids start to drop out at the bottom needle, and we then collect these droplets into suitable containers and make fractions, and after these fractions are analyzed. This approach, however, has some inconveniences. So we, for example, handle infectious samples with sharp hypodermic needles. When we are puncturing the tube, uncontrolled leakage of infectious material around the puncture site appears. The inserting of the needles is dependent on the operator, and also the collection of fractions is dependent on the operator. And nonetheless, when we collect the fractions, it takes quite a long time between the collection of fractions and then analyzing the fractions and reporting the results. So what we did to bypass these limitations, we have developed in our lab an automated approach that mitigates some of the drawbacks of manual sample harvesting. So we are presenting here the PatFix UC sampler that basically automates the entire fraction collection and sample harvesting protocol. So the tubes are inserted into a specifically designed stand that basically minimize all the movement of the gradients. After this, the tubes are automatically penetrated by a set of non-coring, watertight seal providing needles. The operation of the UC sampler is completely controlled by the PatFix software, and the UC sampler, of course, is fully integrated into our existing PatFix hardware. So the chromatography pump empties the content of the centrifuge tube through the series of detectors like UV, fluorescence, and mass, as Andrea was already mentioning before, and this enables chromatography-grade characterization of samples. So after performing this profiling, we obtain so-called centrifugram, which is a density gradient sample-specific profile. Basically, this centrifugram resembles very closely to a chromatogram that we already discussed above. Also here, we are measuring different parameters with different detectors. For example, conductivity monitor is used as a surrogate indicator for cesium chloride gradient density. We measure tryptophan intrinsic fluorescence of proteins from where we can estimate relative areas of empty peaks and full peaks. Light scattering, for example, permits further determination of capsid size and mass, and we use UV absorbance at various wavelengths as let's say general purpose detector. From the evaluation of these profiles, we can extract various information about the sample, and actually the evaluation of these centrifugams is exactly the same, and it is performed with the same workflow and with the same software as the evaluation of chromatograms. So for example, if we integrate the areas, we can calculate the empty to full ratio. If we calculate the ratios between the different detector signals, we can further characterize the type of molecules that are eluting at specific time point in the gradient. So for example, with the ratio 260 and 280, we can quite precisely identify and distinguish between empty and full AAV capsids, for example. So I'm here showing you the profile for the AAV8, but we tested the approach with several AAV serotypes, and it turned out, in contrast to chromatography, that density gradient ultracentrifugation provides serotype-independent separation of AAV capsids in cesium chloride gradients. So the quality of separation between different AAV serotypes is preserved. So for example, if we compare here AAV8 and AAV9, we see that the separation between empty and full peaks is approximately the same. In addition to full and empty capsids, ultracentrifugation indicates heterogeneous populations of various AAV capsids. So not only we can see fulls and empties, but we can see other capsid subtypes like overpacked, which are a bit more denser compared to the fulls, so they are eluting at higher cesium chloride concentrations. We can see partially filled capsids. And in the case of AAV9, we demonstrated that we also can detect various unidentified species that would require further evaluation and characterization. So with the question marks, we see a subpopulation of low density, large particle size components that are, for example, coexisting with the AAV sample in this specific material. Additionally, it turned out that this approach also enables profiling of samples during the downstream processing. So again, the samples were centrifuged and profiled, and from these centrifugograms displayed on this slide, we can actually monitor how each individual downstream process is altering the composition of our samples. So basically, we can monitor the impurity removal from the samples with this ultracentrifugation approach. So on the first centrifugogram on the far left, we see a lot of impurities, and we can barely see the virus. And when we do the TFF and then further clarifications, the virus peaks are starting to be more visible, although we, for example, still see some components that are much more prevalent in the sample, and that they are, for example, concentrated with TFF. And later we see what we are actually removing, for example, with cation exchange capture and elution. So the monitoring of the AAV purification was nicely implemented with this approach, and we were able to see certain components or certain impurities that are not able to be distinguished with traditional chromatography methods or other analytical methods that are used for this type of sample characterization. So we were very happy with the results of this profiling, and the method turned out to be quite interesting for further characterization of AAV samples. So we were, of course, interested how accurately we can actually quantify our samples with this approach. So we performed a similar method validation as Andrea was explaining before for chromatography. Again, we prepared first different concentrations of AAV and estimated the linearity of the method, as well as limit of detection and limit of quantification. And what we observed here is that sensitivity of the method is not as great as, for example, chromatography. So limit of detection and limit of quantification is 1 to 2 orders of magnitude higher compared to chromatography. And this is due to some machinery limitations. But we can see that linearity of linearity of the calibration curves is really great. So the method can serve us as a quantitative method for characterizing AAV capsids. Regarding the LOD and LOQ, I want to mention that still, even though the LODs and LOQs are not as great as with chromatography, this approach enables us much better sensitivity compared to sedimentation velocity ultracentrifugation, analytical ultracentrifugation, where typically at least one order of magnitude more sample is needed to obtain, let's say, relevant signal responses. We continued our validation with testing the interday and intraday reproducibility, and we were actually very surprised how robust the method is. So we determined that UV signal provide excellent robustness with RSD below 1%, which is really, really robust. And we were even more impressed by the fact that when we measured the elution conductivity of the peaks, this RSD was even better. It was below 0.3%. That means that ultracentrifugation in cesium chloride gradients is extremely robust method, and we can generate reproducible results during the centrifugations. Light scattering signal show a bit less accuracy, which is quite obvious because we are working with high refractive index solutions, which are already influencing the light scattering readout. We were a bit surprised by the result of fluorescence detector, which turned out to be not as accurate, for example, as UV or light scattering. And when we studied the scientific literature, it actually turned out that cesium chloride is heavily influencing the fluorescence properties of AAV capsids, and it is generating a lot of background noise. And this, we explained, that is influencing our robustness of the fluorescence detector. However, let's say using UV or light scattering detector, we can still very accurately determine either the AAV concentration or the empty and full AAV ratio in these samples with quite nice sensitivity. So we saw earlier when Andrea already and later myself were showing the calibration curves for different detectors, that there are significant discrepancies in empty and full response between different detectors. So this leads us to a question: which detector enables us to determine the true empty and full AAV ratio? So which detector to trust and to choose if we want to quantify our AAV samples? And I will try to answer this question in the following slides. But before we continue into deeper investigation, I want to point out again the differences in AAV composition. So just to simplify the situation, I will talk about empty and full capsids. So the empty capsids contain only the capsid proteins, whereas with the full capsids, the capsid proteins contain DNA. And if we just have a brief look about the difference and how these differences are reflecting when we measure those two capsids with different signals, For example, a DNA insert increases the UV absorption at 260 nanometers. So usually the full capsids display stronger absorption at 260 nanometers. If we look at the capsid fluorescence, that there is no difference in composition between empty and full capsids, and if we assume that DNA insert is not fluorescence active, that means that there shouldn't be no differences in fluorescence properties. Whereas if we look at the light scattering properties, the light scattering is distinguishing in one side the size of the molecules, on the other side is distinguishing the molecular weight of the molecules. So there should be no differences if we assume that both empty and full capsids have the same diameter, for example, but there is difference in molecular weight because the full capsid contain additional DNA. So again, the full capsid should display stronger light scattering compared to the empty capsids. And indeed, all these properties are reflected into the differences in the slopes of the calibration curves. So here we see that, for example, for the UV detector, full capsids, which are indicated as blue, are displaying stronger UV absorbance compared to the compared to the empty capsids, which are indicated by red lines. Again, light scattering on the bottom calibration curve, full capsids display stronger light scattering compared to the empty AAV capsids. And our assumption about the fluorescence and the fluorescence detection of AAV capsid was actually quite on spot. So because we only measure proteins with the fluorescence detector, we see very little difference between the fluorescence properties of empty and full AAV capsids. So if we go further and actually go back to some absorption basics, so according to the Beer-Lambert law, the absorbance is defined as a sum of extinction coefficient, concentration, and optical length. And based on this assumption, from the slope of the calibration curve, we can actually access some information about the extinction coefficient of our empty and full capsids. And just for the simplicity, I assume that fluorescence and light scattering are behaving similarly. There are some smaller differences, but let's keep it simple during this webinar, so we'll neglect these differences. So as I said, the slope of the calibration curve contains information about the extinction coefficients for the corresponding capsid, and this extinction coefficient is simply telling us how strongly the particle is interacting with the specific wavelength or the specific type of the detection. So the ratio of extinction coefficients therefore indicate the relative responses of individual capsid types. And basically, by calculating these ratios, we are simply looking how much stronger full capsids are, for example, absorbing light or scattering the light compared to the empty capsids. And if we calculate these values, we see that for the UV 230 nanometers, the fulls are absorbing approximately 2 times more light compared to the empties. At 260, because of the strong absorption of nucleic acids, the absorption of fulls is 10 times stronger compared to the absorption of empties. For 280, this absorption is 4.5 times stronger. Fluorescence, it gives us the same absorption, and light scattering is again absorbing light 2 times, scattering light 2 times stronger compared to the empty capsids. So how to prove that this assumption is correct? We did the final experiment. So we purified empties and fulls with ultracentrifugation because it gives really superior separation between the capsids. And after this, we prepared a series of mixtures of empty and full capsids with various empty to full ratios. And we analyzed these mixtures with the chromatography method that Andrea discussed above. When we obtained the chromatograms, we calculated the percentage of full capsids by using 2 approaches. So one approach is the traditional one, where the area of full is divided by the total area under chromatogram. And the second approach was the same formula, but it contained this response factor correction for the empties. And what we see from the graphs that are showed here on this slide is that indeed fluorescence detector is giving us equal response for empties and pools, whereas all other detectors are overestimating the percentage of full capsids. And this overestimate is indicated by the void circles here on the graphs. Whereas if we take into account this response factor, we can basically align the expected and experimental empty to full ratios into a linear form. So basically by implementing the response factor, we can use any detector to accurately quantify empty and full ratio in any AAV sample. So to conclude my part of the presentation, we demonstrated that compared to chromatography, density gradient ultracentrifugation offers an orthogonal, qualitative and quantitative method for the comprehensive characterization of AAV materials, providing insight into sample heterogeneity beyond traditional chromatographic approaches. So we can see with ultracentrifugation things that we cannot see with chromatography. Validation of the post-DGUC profiling approach demonstrated accurate and robust qualification and quantification of empty and full AAV capsid content. So this approach is completely on par with chromatography, for example. Integrating an UC sampler into the PATFIX system enables automated, reliable, and multi-detector supported profiling of post-DGUC samples, which is applicable across all the AAV serotypes. And it's on top of this capable of analyzing samples from various downstream steps without requiring sample pretreatment. It also reduces manual fraction collection errors and improves precision by ensuring accurate sample handling. Later, we saw that discrepancies between detector responses can be addressed by applying empty to full relative response factors, and by incorporating these factors into the percentage of full calculation ensures consistent result across all UV detectors, UV, fluorescence, and light scattering. And finally, this study highlights the potential of DGUC-based profiling to serve as a robust tool for routine AAV characterization and quality control, and contributes to more efficient and reliable manufacturing processes for AAV therapeutics. This work, of course, has not been conducted only by me and Andrea. So at this point, I want to thank my colleagues at Sartorius Bioseparations for their contribution. I want to thank the process analytics development for developing the chromatography methods, the virus process development team for providing us the AAV samples. We did the ultracentrifugation techniques at the basic research, and our product managers helped us significantly with their comments and ideas. And finally, I want to thank Aleš Strancar, our managing director, for all the support and comments. And finally, I want to thank you for your attention.
Brilliant. Thank you very much, Andrea and Sebastian, for those great presentations. So before we start our Q&A, we'd just like to ask the audience a quick polling question, which should be popping up on your screen about now. I'll give you a moment to answer that, and then we'll get started with the Q&A. Okay, I'm going to invite the speakers to join me now on camera, and we will get started with our Q&A. Wonderful. Okay, so first question I've got here is for Sebastian. Given the variability in AAV serotypes and production conditions, how robust is post-DGUC profiling across different AAV variants and manufacturing platforms? Sebastian.
So thank you for this question. So according to our experience working with different AAV serotypes and different production lines, this approach is actually surprisingly robust. So we saw consistently very robust performance of the post-DGUC profiling across different AAV variants and manufacturing platforms.
Brilliant, thank you. One for Andrea here. What advantages does the new AEX method offer compared to other orthogonal analytics for AAV capsids? So things like AUC, CDMS, or cryo-TEM?
Yeah, as I mentioned before, ion exchange chromatography, it's really the well-established technique which demonstrate a really high throughput analytics, and this is one of the advantage of this analytical method. The other thing is the method really it's a linear. We can do linear scalability of this method, which is very key advantage for the purification part, as well as sample amount consumption. Those are.
Brilliant, good answer and good questions coming in. So keep your questions coming in. We'll get to as many as we can, and we'll answer any that we don't get to today by email. So do keep those coming in using the Ask a Question box. Another one here for Andrea. Is it possible to apply the new AEX method across all serotypes?
I mean, we demonstrate different serotypes that this method works, specifically serotype 5, 8. We tested also serotype 2, and we see quite good results also, or good indication also for AAV9 serotype.
A good range. And Sebastian, how do you see the role of post-DGUC profiling evolving in routine AAV characterization workflows? Could it eventually replace or maybe more complement chromatography-based methods?
So the post-DGUC profiling is indeed very interesting approach. And I think it complements the established characterization methods quite well. So I think it's not really competing with chromatography, for example, but it's nicely complementing it. It perhaps competes more with sedimentation velocity, AUC, which was a workhorse for empty full characterization for quite a while, but there are certain limitations that people want to avoid. So even in the field of AUC, the people are moving towards the density gradient equilibrium profiling, which is basically the approach that we implemented with our approach. However, with our approach, no AUC equipment is required. So we can do this profiling on regular laboratory ultracentrifuges, and the characterization of samples and evaluation of the data is very straightforward. So here I see more competition, and our approach, I believe, is much more straightforward and easier to use. And it complements really great to other characterization techniques like AEX or mass photometry or scanning electron microscopy and so on.
Brilliant, thank you. Another one for you, Sebastian. How do response factor corrections improve the reliability of empty-full quantification across different detection modes? Could this approach be applied to other AAV analytical methods beyond DGUC?
So this response factor correction is actually enabling us to take any detector that was traditionally used for characterization and align the responses between those different detectors. We used 3 detectors in a row in our approach, but many times people are using just 1 detector. So basically, by implementing this correction factor, you can obtain the right empty to full ratios from any detector without compromising the accuracy. Of course, you have to determine this correction factor in advance, and you have to determine this correction factor in advance for your specific serotype and for your specific DNA content. So when we compare different serotypes, we saw that there are slight differences between the correction factors for individual AAVs. So these here are reported only for AAV 8, and for example, for AAV 9, they are a bit different. And again, if you change the insert, the correction factor will again change. So this has to be done individually for, let's say, user-specific AAVs.
Brilliant, thank you. Andrea, what types of samples can be analysed? For example, crude or in-process samples, and is any sample preparation required before QA analysis?
Specifically, good question. Specifically for QC method, absolutely, purified samples are favorable. But however, we also tested some crude samples. And really, you get some kind of indication of empty and full separation. However, you will need to use, for that kind of crude samples, you will need to use also light scattering detector. With the method, normally it's just dilution with the mobile phase A, but otherwise, analytics is just straightforward.
Perfect. And could you please explain if the empty-full relative response across the different detection wavelengths or detection methods was measured in cesium chloride, and how do these responses change in cesium chloride versus sodium chloride?
So the correction factors were actually determined based on the post-DGUC profiling in cesium chloride, simply because we have superior empty to full separation, and we can obtain very precise information about the content of empty and full. But later, these same factors were used to characterize the samples that were measured with AEX in sodium chloride gradient. So we were able to demonstrate that these correction factors are valid for specific serotype and for specific insert independently on the media where these AAVs are. So it is the same for cesium chloride as well as for the sodium chloride or for any other mobile phase where these viruses are currently. Probably there are some minor differences, of course, because the solvent is affecting, for example, fluorescence properties of the proteins, but we were not able to distinguish these differences within our experiments.
Okay, brilliant. Well, thank you very much, Sebastian and Andrea, for answering those questions. That is all we've got time for today. So any questions that we didn't get to, we're going to try and reply to those by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with some feedback on today's webinar. The link to that can be found in the resources on the right-hand side of the screen, and it will also be in the on-demand email. Additionally, there's an attendance certificate that is available also on that right-hand side. So all that's left then is to thank Andrea and Sebastian once more for a great presentation, and thank you to the audience for watching. We do hope you join us again soon.